In this section, I'm going to talk about an introduction to adaptive immunity. So before we talk about adaptive immunity, we need to go through lymphocyte differentiation and development, so you have an understanding of what these cells are capable of. So we start with lymphocyte differentiation. So when your cells are being created, we all start with stem cells, and stem cells can typically become every cell in the body. Especially if they are embryonic stem cells, they can become any cell in the body. And when stem cells differentiate a little bit further, they can become a specific type of stem cell. And one type is called a mesenchymal stem cell. With mesenchymal stem cells, these are committed to becoming a small subset of cellular types. They can become neurons and neuroglial cells. They can become fat cells or adipocytes. They can become osteocytes or bone cells. So progenitor cells can become osteocytes and osteoblasts and so forth. And they can become hematopoietic stem cells. Hematopoietic stem cells is a specific type of stem cell that can become any blood cell in the body. So they can turn into red blood cells or white blood cells. And from here, there's different lineages that they can go on. But eventually, so there's the red blood cell lineage, the white blood cell lineage, and then from white blood cells, they either become the granulocytes or the monocytic or the lymphocytic. And from the lymphocytic, they can become a variety of lymphocytes, so natural killer cells, T cells, B cells. And we're going to focus on T cells and B cells. T cells are going to make up your cell-mediated immunity. And B cells are going to make up your humoral immunity. T cells are going to go on to develop in the thymus, while B cells are going to go on to develop in the bone marrow. So we talked about the differentiation, and now we have to talk about the development. So when the T cells go to the thymus and when the B cells go to the bone marrow, they obtain a specific receptor. So if you remember from my previous lecture about the innate versus adaptive immune response, in innate, it's nonspecific. It doesn't recognize what it's attacking. It just attacks anything that's abnormal or foreign, while adaptive specifically recognizes the substance that's foreign or the substance that's causing a problem. So with adaptive immunity, we have to have a specific receptor on the surface in order to do that recognition. For T cells, it's going to be a T cell receptor. And for B cells, it's going to be an antibody. So I'm going to switch over here, and I'm going to add a slide so that I can draw a little bit easier. So now we have I'll change the color here. That's good. So we talked about T cell receptors and antibodies. So if we draw a picture of a typical cell, there's a special receptor on the surface of that cell. And that receptor's job is to recognize foreign antigens. These receptors are randomly made, so we have this clonal selection theory. A theory is a well-tested hypothesis, so we're pretty sure that this is the case, but it's not necessarily a law. We don't know it 100%, but we're pretty certain that this is what happens. So we are pretty sure that these receptors are randomly made and recombined to take on a whole bunch of different shapes. And each one of these receptors is designed to recognize a specific antigen. So when we make a T cell or a B cell, we're actually making cells that have different receptors on them. So this one may look like this. We may have one that looks like this, and one that looks like this. Now, since we've randomly created all these different types of receptors, there's a chance that one of these receptors may recognize our own cells as being foreign. Well, we need to select against those. We need to get rid of them. So the clonal part is making tons and tons of different copies with all different types of receptors on them. The selection part means that we have to select against or kill off the ones that recognize our own self as being foreign. So in the thymus for T cells or in the bone marrow for B cells, we have self cells. And what they're going to do is you have a self cell, and your self cell will have self antigen on it. Each one of these T cells is going to be tested. These are all T cells, T cells, T cells. So this one is going to bring its receptor near the self cell. Since the receptor is the wrong shape, it's not going to bind. This one passes the test, it's free to go. This one is going to bring its receptor toward the self cell. Because the receptor doesn't match the surface here, the surface marker or the antigen, this cell is past the test and it's free to go. This one is going to come up to the self cell, and if you look at the shape, this shape fits the shape that's on the surface, the self-antigen shape. There is a likelihood that this cell will recognize our own cells as being foreign, so we kill off this cell. Now this one comes up to it, and it too is going to present its receptor to the self-antigen, and if it doesn't bind and doesn't recognize self as being foreign, then it's allowed to go and reproduce and go hide out in the lymphatic system until it gets activated, until somebody calls the cousins for backup. So in the clonal selection theory, numerous types of lymphocytes are produced with different types of receptors, and we select against or kill off those receptors that will recognize and bind to self-antigen, because we don't want immune system cells floating around in our body that think that we are the bad guy. Presentation of antigens. So there's 3 different ways that our lymphocytes can be presented with antigens. So if we want to wake up this adaptive immune response, there's 3 main ways to do it. The first way is probably the most obvious, would be on the pathogen itself. So we have antigen on the surface of the pathogen, and that's the most direct way to stimulate the lymphocytes. The only problem with this one is lymphocytes typically are in very small concentrations in the blood and even smaller concentrations in the tissues. They hang out in the lymphatic system predominantly. So if you were to get a paper cut and your lymphatic system were to suck up the fluid and suck up some bacteria out of that paper cut, that fluid would then be carried to your lymph nodes to be cleaned out and likely come in contact with lymphocytes in the lymph nodes. If the bacteria got into your bloodstream, the bacteria would be carried through the bloodstream and taken to the white matter of the spleen, and it would come in contact with lymphocytes there. But if this was an infection of the digestive tract, and there's no direct drainage of lymphatic fluid or no blood vessel involvement here, the chances of this pathogen actually coming in contact with your lymphocytes is pretty rare. So there has to be another way to wake up your third line of defense or to call the cousins for backup. So on the pathogen itself is one way. Another way would be on an antigen presenting cell. So we have an antigen presenting cell, and the antigen presenting cell is going to hold it up, and I'll abbreviate that APC. And this is a receptor called MHC class 2, which we will talk about more later on. MHC class 2 is capable of holding up antigen and presenting it to the immune system. If you've ever seen those old war movies where there's people riding around on horseback and there's one troop of soldiers that's trying to do a sneak attack on this other troop. So the guy on the horseback rides over to them and kind of like chops one of their heads off and then runs back to town saying, hey, they're coming to attack us, and starts like alerting everybody that we're under attack by these terrible things. That's kind of what antigen presenting cells do. So what they'll do is they will eat a foreign bacterium or a foreign substance, and then they're going to hold only a chunk of it up to your body. So they're going to eat it and internalize it and take a small chunk of it and hold it up. Like, hey, this is what I just ate. And these antigen presenting cells are able to float around the body. And they can go through the bloodstream into the lymphatic system all over the place. And they are going to seek out the lymphocytes to activate those lymphocytes. So antigen presenting cells can then move these around in the body and go all over the place looking for those lymphocytes. Antigen presenting cells include macrophages, dendritic cells, B cells, amongst others. So these are just some pretty common ones. They can internalize the pathogen, rip off a chunk of it, hold it up on this receptor, this little arm called MHC class 2, and then they can move around the body trying to wake up the immune system to attack that substance. The other way is on the surface of MHC class 1 of an infected self cell. So you have this infected self cell. And the infected self cell has something inside of it, typically a virus, and there's some intracellular bacteria as well, and other types of pathogens that can live out inside your cells, but a lot of times it's going to be a virus. So let's say that there's a virus inside of here. The infected self cell can hold up part of the pathogen on this arm called MHC class 1. And when it holds it up on MHC class 1, a cell that recognizes this situation will come along, and if it's a lymphocyte, like a cytotoxic T cell, it's going to recognize this situation, and it's going to kill the infected self cell. Now, why is it going to kill the infected self cell? Well, there's infectious particles hiding inside of this cell. So in order to clear the body, the multicellular body of this pathogen, the immune system will kill the one cell and sacrifice that one cell in order to release all those particles so that they can't later go on and infect other cells. Once this cell is killed, it ruptures and all those infectious particles will be released, and then the immune system cells can kind of gobble them up. Kind of like if you ever had kids that did something like naughty and you're trying to yell at your kids and you can't find them, and all the kids happen to run inside of a tent, and now they're hiding inside of a tent. And all you see is their little foot sticking out of this tent. So you go up to the tent, you can see the foot sticking out, you know all the kids are in there, and you know that they've done something naughty. So what you do is you rip the blanket off the top of the tent, all the little kids come running out, and you can punish them accordingly for doing whatever they did. That's kind of what the immune system will do. The immune system sees a component of this pathogen sticking out of the infected self cell, and it's going to kill the infected self cell, releasing the particles so that they can destroy the remaining particles, so that those particles can't go infect other cells. So the next thing that's going to happen is T cells and B cells are challenged. So you have the lymphatic system, and I'm just going to talk about a lymph node, but there's all sorts of different types of lymphatic tissues, and we talked a little bit about them in the review of the lymphatic system. So if we have a lymph node here, the lymph node is kind of circular. There's some, the cortex region, and then you have these medullary cords in here. And all in all, it's kind of like a meshwork. And this meshwork contains T cells and B cells. And the reason why it's a meshwork, and there's different layers to it, is to slow the flow through the lymph node. If things were to move too quickly through the lymph node, then you wouldn't have an adequate chance for the pathogen to come in contact with its lymphocyte. So we've got T cells and B cells inside of this lymph node, and they're kind of held up in a meshwork. So now we're going to bring lymphatic fluid through the lymphatic system. So in this example, we're going to bring it through a lymph node. When the fluid comes in, the fluid is going to slosh around the cortex, and then it's going to eventually end up in the medullary cavity here in the medulla, of the lymph node. And it's hoping to come in contact with the 1 or 2 lymphocytes that are able to recognize the pathogen as being foreign. So if this lymphatic fluid contained pathogens, so little bacterial cells, these are going to be carried into the lymph node, and maybe only one of these cells is going to be able to recognize that pathogen as being foreign. So because each one has a different receptor and each receptor recognizes a different pathogen, there's likely that only 1 or 2 of these cells can actually recognize this pathogen. So when the fluid comes in, like I said, it sloshes around in hopes to find that one that it can activate. Once bound, that's what leads to B cell or T cell activation, which we'll talk about in a little bit. Think about walking into a party. Let's say you got a phone call from a friend and said, hey, meet me at this party, and it's wall-to-wall people, and it's really difficult to navigate through this area. It's one large room, and there's people everywhere. You're going to bounce through all of those people, and you're going to be looking and looking and looking until you come in contact with your friend. And when you meet your friend, you're going to say, hey, I'm here, and you're going to have fun together. So that's kind of what it's like inside of a lymph node. These bacterial cells don't necessarily want to see their attacker, but they're going to be bouncing around inside the lymph node and come in contact with a bunch of different receptors. And these receptors are all going to be waiting for their pathogen to come into that lymph node. If that receptor recognizes that pathogen, they're going to bind, and that's going to stimulate activation of the T cell or B cell. Some of these T cells and B cells in your lymphatic system will be waiting your entire life or their entire life for the correct pathogen to come in. Let's say this B cell over here recognizes the antigens that are only found on Yersinia pestis. Well, Yersinia pestis causes the bubonic plague. The likelihood of you getting bubonic plague in your lifetime is pretty slim. So there's a chance that this cell will never be activated because the only thing on earth that can activate it are the antigens of the bubonic plague. Now, if they happen to develop a vaccine against the bubonic plague, and you happen to get that vaccine will be able to stimulate this cell and produce a slight immune response to confer that immunological memory, that benefit of the vaccine. But it either has to come in contact with the pathogen, a chunk of the pathogen, or a vaccine in order to be stimulated. And if you never come in contact with it, this cell will never be activated. But this cell over here, let's say it recognizes Streptococcus pyogenes, group A strep that causes strep throat. This one is likely to get activated in your lifetime. If you think, have I ever gotten strep throat before? Probably. So that cell is there waiting to recognize the antigens present on strep. You have another cell nearby that can recognize E. coli, another cell that can recognize Borrelia. So each one of these cells is very specific. And that's what is meant by the specific immune response. It's specific because the receptors, either the T cell receptor, if it's a T cell, or the antibody, if it's a B cell, are able to recognize a specific antigen. And they are only activated when that receptor interacts with that specific antigen. So that's why we call this specific, and it's the adaptive immune response versus innate, where innate, anything that's foreign, will stimulate the response.